Hi, my name is Natalie, and our topic is water reabsorption and the loop of Henle. We'll be talking about the proximal convoluted tubule, the vasa recta, the descending and ascending limb of the loop of Henle, the distal convoluted tubule, and the collecting duct. Long nephron loops of juxtamedullary nephrons create the concentration gradient, and as water and solutes are reabsorbed, the loop first concentrates the filtrate and then dilutes it. The vasa recta will preserve the gradient, and then the collecting duct will use the gradient to determine the final concentration and volume of urine.
Hi, my name is Jamie, and I will be talking about the proximal convoluted tubule. The wall of the PCT are formed by cuboidal epithelial cells. This contains microvilli and brush border for the absorption of water and molecules. The solutes that are reabsorbed include glucose, amino acids, and 65% of salt and water. These salts require ATP to be reabsorbed via active transport.
Next, we're talking about water reabsorption in the nephron loop, also known as the loop of Henle. It is responsible for concentrating or diluting the tubular fluid using energy from the kidneys through a process called countercurrent multiplication. This is a process that uses energy to generate an osmotic gradient that enables you to reabsorb water from the tubular fluid and produce concentrated urine. That's where the vasa recta comes in with the concentration gradient. Next, we'll move on to activity in the descending limb. So this is the thin part of the loop of Henle, and it's made of simple squamous epithelium. It receives isotonic fluid from the proximal convoluted tubule. It's isotonic because as ions are reabsorbed by the gradient, water is also reabsorbed, maintaining the osmolarity of the fluid in the proximal convoluted tubule. Permeability here to water is high, and there's a low permeability to ions and urea. Water is easily reabsorbed here, and solutes are not, and filtrate becomes concentrated because of this. Fluid from the loop of Henle loses water to the higher concentration outside the loop and increases in toxicity until it reaches its maximum at the bottom of the loop. That means that this area represents where the fluid has the highest concentration in the nephron.
Hi, I'm Kim, and I'll be talking about the activity that takes place in the next portion of the loop of Henle, which is the ascending limb. The ascending limb is also known as the diluting segment of the nephron because of its ability to dilute the fluid in the loop. Its walls consist of thick cuboidal cells, and solute sodium and chlorine are actively pumped out of the ascending limb into the intercellular parts of the kidney medulla. This process is maintained by the 3-ion cotransporter, which consists of sodium, potassium, and chloride, and the sodium-potassium ATPase. The ascending limb contains a lower amount of fluid than the descending limb does because there's no water absorption taking place here due to the water impermeability of the wall. This makes this part of the loop of Henle less concentrated than the descending limb. The loop of Henle continues to become less concentrated by solutes as you move up towards the distal convoluted tubule, eventually becoming hypoosmotic to the interstitial fluid at the top of the ascending limb.
Next is the distal convoluted tubule. The DCT is responsible for the reabsorption of water and homeostatic process. The walls are lined with simple cuboidal cells. This regulates pH by absorbing bicarbonate and secreting hydrogen ions into the filtrate. Sodium absorption is regulated by aldosterone. This then leads into the collecting duct, which is the final portion of the nephron where 5% of reabsorption occurs. ADH regulates the amount of water reabsorption in the collecting duct. The collecting duct becomes more permeable to H2O when ADH is present, resulting in an increase of H2O reabsorption. ADH increases the amount of aquaporins. These aquaporins are integral membrane proteins that act as water channels, playing an important role in water balance. The remaining solutes and liquids present become urine.